A major focus of my lab is understanding the mechanical properties of biological materials, and that ranges from animal cells to plant cells, and we're particularly interested in understanding the cell nucleus, which is where most of the genomic material, most of the DNA of the cells is contained. So there are many cells that appear in this way, such as cells in our blood, white blood cells. In some cases, however, the nucleus has a different shape. So this is characteristic of some types of cancer cells where this increased volume it's occupying inside of the cell is actually used as a marker for diagnosing and even prognosing cancer. So some types of breast cancer, for example, the more severe this shape irregularity, the worse the prognosis for breast cancer. This matters for disease. So cancer cells, for example, are softer, more flexible than benign cells. And in fact, when treated with different drugs, they become stiffer. So why is that? This is a good question, something that we're trying to better understand. In order to do this, we're building a lot of new technologies to be able to probe the texture or squishiness of individual cells and nuclei. How do we probe the mechanical properties of cells? These are very small objects, typically 10 microns or 20 microns, maybe more. This requires using very small pieces of equipment. One of the ways we do that is we make a tortuous path fabricated into some silicon polymer, commonly known as a microfluidic device. And then we force the cells to squeeze through these narrow constrictions, which gives us some indication of the deformability of the whole cell. We image the cells and the deformations of them using microscopes and high-speed cameras. So some of these technologies that we're developing could have eventually a clinical impact. They could be used by doctors to be able to diagnose or even determine which drug treatment might be most effective for a particular patient. These are, of course, much longer-term potential applications, but it's something that another dimension of our work that we're excited about.
There's all this cultural history and value locked in this collection, and what we try to do in Access is try to help people unlock that. In a collection this large, you have material on basically every major topic relevant to the history of the 20th century. We have over 300,000 holdings, and the collection spans from the dawn of cinema to the present. So we have a lot of everything.